We are in for a real treat today. I am so pleased to welcome Dr. Livia Eberlin to our Canary Center at Stanford for Cancer Early Detection seminar series. Livia received her bachelor's degree in chemistry from the State University of Campinas in Sao Paulo, Brazil. She followed that with a PhD in analytical chemistry at Purdue with Dr. Graham Cooks. So technically she is my academic aunt, even though I'm much older than she is. So she then came to Stanford to do her postdoc in chemistry with Dick Zare, and from there moved to UT Austin to start her independent faculty career. And a couple of years ago now, she moved to Baylor College of Medicine to really immerse herself with surgeons and in a more medical environment, which has been really exciting, I think, for a lot of her research. She is currently an associate professor and translational research and innovations endowed chair in the Department of Surgery. Professor Eberlin is recognized for her innovative research in analytical chemistry, medical mass spectrometry, and cancer research. She also has this really long list of honors for her research. I'll name a few of them here: an NIH K99 R00 Pathway to Independence Award, Forbes 30 Under 30 listing in healthcare, Sloan Fellowship, Moore Inventor Fellowship, and a MacArthur Fellowship. So very exciting. Her research program centers around the development and application of novel mass spectrometry technologies for health-related research, with a particular focus on disease detection and diagnostics. She is also notably very passionate about mentorship, scientific education, and efforts related to diversity and inclusion within the scientific community. So I'll also just end with a little story about Livia. She and I first met when I was a brand new assistant professor at Stanford. Canary Center was back on California Avenue back in the days. And she shows up in my office like carrying this DESI source. And she's like, "Hi, I just need the instrument to put this on. Are you using your instrument full time? Can I come use it at night?" And I was just blown away because here's this postdoc with not all the resources to do what she needs to do. And she was determined to get them. And she did amazing things at Stanford. And it's just been such a pleasure to watch her career and all the incredible things she's accomplished in bringing mass spectrometry into the operating room, which she'll tell you about today. So welcome, Livia.
Did I do the same thing? Yeah, well, I remember, Rob, that very well, because I remember getting data and going to Dick there and just being like, we need someone to do statistics. And Dick just stood up and he said, "I know who, and let's walk through Rob Tibshirani." So that really changed my life. So good moments. Thank you so much, Sharon. Thank you all for coming. It's my pleasure to be talking about some of my research program today. If you saw my talk yesterday, there will be some overlap, but I'll also show you a little bit more of what we do on the cancer research side of things. I'm primarily at Baylor College of Medicine, but I have an appointment at MD Anderson as well, and have loved being in the Texas Medical Center, and it really gave me this awesome opportunity to be close to surgeons, pathologists, but most importantly, too, being close to patients. And that's made a huge difference in our ability to get patient samples, being part of some incredible clinical studies, and really digging deep into the translational research. So I just want to take 1 second to thank Stanford, this incredible institution, for having me here as a postdoc. That was really a changing moment in my career. I want to thank Dick Zare, who was my advisor, for giving me the opportunity to be here. That's the DESI source that Sharon was telling you about. And yeah, at first we were really trying to get any and all instrument time that I could. I want to thank Rob, too, who's been an incredible mentor to me in many different ways, and still a collaborator in our projects. And a bunch of my friends that I made here that have become colleagues and just really lifelong partners in life and in science. So really an incredible time that I had here at Stanford. This is my group at Baylor College of Medicine. We're right at the heart of the TMC. We're at Baylor. That's where my lab is. We have our mass packs there, but we also have 3 systems that are deployed. I like to say they're in the operating room, and I'll show you some of our OR work today, but it's been just a pleasure being there and as I said, close to the clinicians. A lot of people ask me, why did you move from chemistry to surgery? I am a chemist. I'm an analytical chemist. But I moved to surgery, again, to be close to patients and to be in the TMC. When I was at UT Austin, I was already having projects in collaboration with MD Anderson and Baylor College of Medicine. We were going to Houston all the time. And the main thing that was driving this is really access to patients. So at the TMC, we treat about 10 million patients every single year. So just to give you a perspective of the incredible opportunity there is to conduct clinical and cancer research that can make an impact in the lives of these patients, and just being close to that medical community and the enthusiasm that there is it's been really awesome. So I'll tell you about what we love to do in my lab, which is what I like to call medical mass spectrometry. You may have heard about clinical mass spectrometry or proteomics, lipidomics, metabolomics that all use different forms of mass spec. In my lab, we like to think about mass spec as a potential new medical technology. Not a lot of people think of mass spectrometry in that way. But what we want to be able to do is to enable medical doctors and people in the clinic to directly access the power of mass spectrometry technologies to help them drive and make better clinical decisions for their patients. In my lab, we like to do direct mass spectrometry analysis, meaning that we don't really do separations or chromatography. We're taking samples as they are. A lot of times the sample is still within the patient. A lot of the times they're freshly excised from the patients or a biopsy, and directly analyzing them. We like doing mass spectrometry imaging too, and I'll show you some of our DESI imaging work today. But we want to be able to use methods that are quick, that are simple to use, that can be implemented on a routine basis to help expedite clinical decisions. We use a lot of high-performance mass spectrometry in my lab. We love using Orbitrap systems, and I'll show you why high-resolution, high-mass accuracy mass spectrometry data is so important in our research. But we also like our instruments to be smaller and movable. And I'm not talking like super, super small, but things that can be put on a cart and roll around. Actually, I was looking at this cart here. That's like perfect for a mass spectrometer to be rolled around. And the reason for that is that we want to bring mass spec as close as we can to the medical professionals. And we also do a lot with advanced data analysis system, AI and machine learning. We're not experts, so we collaborate with people like Rob and others in our community to make the best that we can of the data that we acquire. We are developing and acquiring really complex data sets, and we need to be able to be very good at interpreting the data and finding what's really significant and what's really important in some of the biological questions that we're asking, but also to derive clinical results. So our goal is to be able to acquire some deep molecular information using mass spectrometry alone, and also convert that complex molecular data into actionable results. And I really like to make that a strong point, because this part where you translate deep molecular data into things that can be interpreted by a medical professional, I think is very important in being able to make an impact, and in also being able to incorporate our technology into routine clinical assays. And of course, in that process, we also are able to make new molecular discoveries. And a lot of the projects that I'll tell you about today, we are also able to identify new targets for treatment in a variety of projects related to patient care. So the idea is that we would really expand the footprint of mass spectrometry to improve patient care. My lab is now in a hospital, which it used to be like in a chemistry building, but now, you know, I have a lab and a research lab, a laboratory in the hospital system, and we do see a lot of mass specs in research labs. There are also a lot of mass spectrometers already in clinical laboratories and a variety of different clinical assays. We've been really trying to push to get mass spectrometers in the pathology room to help with a lot of diagnostic decisions and tissue analysis. And I'll show you also the work that we've been doing bringing mass spectrometry into the operating room. Collectively, the work in my lab is primarily focused on tissue analysis. And in particular, we're really interested in doing metabolic analysis of tissue. So in the next few slides, I'm just going to try to make a point. Why do I think that metabolic analysis of tissues can really have a huge impact in patient care? So when I talk to a lot of my colleagues, which are now surgeons and pathologists, when I say, okay, we want to do molecular-based diagnosis, the first thing that I see comes to their mind are like sequencing, right? They're thinking, okay, molecules are like DNA and RNA and proteins. It's rarely that they think, oh, molecular-based diagnosis can be done with metabolite and lipid analysis. And I think that as scientists, we have made a really good job and really great progress in incorporating DNA analysis, RNA analysis directly of tissue and clinical samples into the routine clinical workflow. So of course, sequencing, qPCR is just standard of care now. Any patient that comes in, they will have, their samples will probably be analyzed with one of these techniques. In terms of protein analysis, proteomic is making its way, right, Sharon, to clinical practice. But if you think about protein analysis, primarily the assays that are being done are like Western, IHC, and ELISA that are being used in patient care. But I think there's still a huge opportunity is actually in directly measuring metabolites from tissues and clinical samples to help with clinical decision-making and understanding also disease processes. And one thing that's really amazing about metabolites and why I'm so passionate about this class of molecules is because from the perspective of chemical diversity, metabolites have a huge diversity in structure in different classes of molecules that can be picked up with mass spectrometry and characterized with mass spectrometry in a really powerful way. And from a biological and chemical perspective, I like to think about metabolites as like the end products of the chemical reactions that are happening in our body. They're being controlled through gene and protein expression. But if we're measuring metabolic changes and the changes in the abundance of specific small molecules and lipids and also sugars, that can really represent like a functional readout, what's actually happening in your cells and in your system right now. And what I hope to show you today is that we can then correlate this measurement of the metabolic composition of tissues with disease diagnosis. We can infer prognosis for patients. We can give information about potential response to treatment options. And I'll show you some projects today with immunotherapy and chemotherapy, and also look into disease development, aggressiveness of different tumors. And what's really awesome about, in particular with mass spectrometry imaging, is that we can use mass spec imaging to look at the distribution of these metabolites within the tumor microenvironment and understand the factor, understand what are the processes related to the stroma, necrosis, and other processes that are happening related to the tumor cells in the patient tissue. The other thing that is great about metabolic analysis is that mass spectrometry provides really incredible analytical performance to directly analyze metabolites from tissues. And one of the reasons that we can do that is that with mass spectrometry, we're measuring the mass to charge ratio of different molecules and their abundances. And our systems have gotten so good, our mass spectrometers nowadays, they're so powerful that when I started in mass spec almost 20 years ago, we wouldn't really be able to separate these two different metabolites that have the same nominal mass. But today we routinely run our mass spectrometers at 500,000 resolving power. We have accuracies of over 5 ppm to the point that we can directly get this data from tissue without chromatographic separation and really know that we're detecting these molecules. We can do 10 in mass spectrometry and know exactly what their chemical structure is. There have been a lot of developments in lipid analysis where we can know how many carbons are in each fatty acid chain, what are the position of the double bonds. So we can really dig deep into this metabolic information, even if we're not doing chromatographic separation and we're just getting profiles of these molecules directly from tissue. So for example, this is a typical mass spectra that we acquired with an Orbitrap mass spectrometer in my lab directly from a tissue. And if you just start digging deeper into the mass spectra, you'll see that the molecular information is really rich. And we can separate molecules that have the same nominal mass, but different accurate mass. And then, as I said, further characterize their chemical structure. So in our analysis, we tend to focus on, in my lab, we tend to focus our analysis on small molecules. We can run our mass spectrometers in both the negative ion mode and the positive ion mode. We really like running things in the negative ion mode, which is not very common for most people that think about proteomics. And the reason for that is that we can really cover a broader range of classes of molecules in the negative ion mode. These are all weak acids that are easy to detect and ionize in the negative ion mode. So we see a variety of small metabolites in the low MZ range, and I'll show you some specific structures and molecules that we're interested in. See a lot of free fatty acids, prostaglandins, fatty acid dimers, stereolipids, cholesterol, cholesterol derivatives, lysolipids. And then in the higher mass range from about 800 to 12 to 1500, we detect more of the complex lipids that are part of our membrane structures, also signaling molecules, nuclear membrane, mitochondrial membrane, and so on. So these are some of the classes of the lipids that we detect. We also run our analysis, and we can do that on the same tissue in the positive ion mode. In the positive mode, our mass spectra is normally composed primarily of diacylglycerides, sphingomyelins, glycerolphosphocholines, a really high abundance, and also triacylglycerides. I get asked often, do you detect proteins as well? We have done some work with direct mass spec with DeBakey, looking at protein analysis if you're interested in these studies, but we really try, and we're more excited about small molecule analysis in my lab. So you may be asking, okay, so how do you actually access this metabolic information? How do you get to obtain these metabolites directly from tissues? And I have to tell you, there are many different flavors of mass spectrometry. There's a class of ionization techniques in mass spec that we call ambient ionization mass spectrometry. So you can do that using lasers, different desorption techniques, and different technologies. In my lab, we like employing what we call liquid solid extraction based ambient mass spectrometry techniques. We've been working a lot with DeBakey, which Sharon mentioned to you. I was doing some of this work here at Stanford as well. I'll explain exactly how DeBakey works in a couple of slides. But DeBakey is really incredible to get spatial information and do mass spectrometry imaging of a variety of different samples, but especially from tissue sections that are deposited on glass slides. We also do DeBakey of fine needle aspiration biopsies of this is actually a tissue smear that we just smeared on the slide, and we can do DeBakey imaging. We're typically running with DeBakey at a resolution of 20 to 250 micrometers. The Waters, who's now selling these systems, they have shown that they can go down to 1 micrometer with DeBakey, which is really amazing for the technology. And then there are other techniques like MALDI and SIMS that provide even higher spatial resolution. Then the other technique that we developed in my lab and that we love using intraoperatively, and I'll show you more about it later too, is what we call the mass spec pen technology. It's not a pen, but it's like a pen. It's a handheld gadget that we provide surgeons and pathologists so that they can analyze tissues that are freshly excised from the patients, but also in vivo. And I'll tell you more about that a little later. But let's start with DeBakey. So DeBakey, it's a variant of electrospray ionization. That's desorption electrospray ionization. What we do in DeBakey is we actually use a spray of charged micro droplets that is directed towards a tissue section or a sample surface. And then these micro droplets, they're about 5 micrometers, traveling 100 meters per second. They will impact this sample surface and then extract molecules from this sample. And then the droplets now containing the extracted molecules are directed towards the mass spectrometer, where they're vaporized and ionized. The molecules are ionized, and then these gas phase ions go into the mass spectrometer. DeBakey is really awesome because you can put this sample in like a moving stage, and then you can acquire mass spectra for every single pixel or position that you analyze. So let's say we get a mass spectrum for every position in the tissue that we analyze, and these are just some examples of the molecules that we detect, like ascorbate, fatty acid, oleic acid, and the glycerophosphoetanolamine. And then if you do this sequentially for the different positions in your tissue section, you get this huge amount of data that you can now put together and display as two-dimensional ion images. Just showing you some examples here of the ion images and the different spatial distribution of different molecules in these tissues. One of the really cool thing about DeBakey is that you can play with the chemistry of your solvent system. And we do that a lot. And we still do a lot of optimization in terms of the chemical composition of the spray. But one of the main things is that you can use solvents that we say that they're histologically compatible, meaning that we do the extraction of the metabolites and fatty acids and the lipid species, but the histology of the tissue remains intact. So we can still do, for example, this is an H&E stain of the exact same tissue that we got the molecular images. So you can do H&E staining or immunohistochemistry. We've been doing immunofluorescence. Recently, we've been doing Visium for spatial transcriptomics. So now you can have that perfect overlay of the metabolic information, even protein information and RNA information on the exact same tissue section. So it's really powerful in that way to do mass spectrometry imaging and molecular analysis of tissues. So one of the first projects that I wanted to tell you about today that we're using DESI imaging, and this is a really incredible collaboration that I have at MD Anderson with JJ Gao and Linhua Wang. These are two people that are very interested in looking at 9p21 loss in tumors and how that correlates to response to immunotherapy and the immune microenvironment of tumor tissues. We got engaged in this process because one of the enzymes and one of the genes that are part of this chromosomal deletion or this chromosomal locus, which is the MTAP protein, has strong implications in metabolism. So this is really where we got engaged into looking at the metabolic composition of these tumors and trying to understand what's happening from a metabolic perspective. So what JJ Gao and Linhua Wang did was they profiled a variety of tumors in this large study at MD Anderson, and they found that the homozygous deletion of the chromosome 9p21 locus is a very frequent genetic defect, genome defect that occurs in about 14% of all cancers. And consequentially, the MTAP co-deletion occurs in about 80 to 90% of these tumors. Clinically, what JJ and Linhua found was that these tumors that have this 9p21 loss, they have what they call a cold tumor immune phenotype. And what they've seen from these patients is that they exhibit a much lower response to immune checkpoint therapy, and they have worse outcomes as well to these therapies. So you can see here some of the data that they published in this Nature Communications paper. And some of the tumors that we see a high occurrence of this 9p21 loss is mesothelioma, bladder cancer, glioblastomas, and also pancreatic cancer. So we looked into what would be the effect of losing, right, of this co-deletion of MTAP, and could we then use the consequences of this co-deletion or lacking this enzyme to detect metabolites that are in these pathways with DESI to be able to identify 9p21 loss and also be able then to provide information in regards to the potential treatment response for these patients. So this is really where our hypothesis came from. We hypothesized that losing this MTAP activity will lead to accumulation of MTA. MTA is 5-methylthioadenosine. You can see the structure here, this really beautiful metabolite. MTA is it's a weak base. Here you can see the amine group. We detected in the positive ion mode at MZ 298.096. And the thought was that if, I don't think I have a pointer. Do I? I don't think so. I'll just show you here. The thought was that if we lose the ability to produce MTAP, we expected to see accumulation of the MTA metabolite in these tumors. So we thought, okay, how do we actually get to probe that? So we worked with JJ Gao in his lab, and they have several tumor xenografts that they have developed in which they actually knocked out the MTAP protein. And Meredith, my student, she spent an enormous amount of time optimizing the DESI system so that we could actually detect MTA directly from tissue. One thing that I've learned now working with DESI for so many years is that the optimization of the system can actually lead to the detection of molecules that you wouldn't think would be possible to be detected before. So MTA is hard to detect, but she optimized the method both in negative ion mode and positive ion mode to detect it. And what we saw was really beautiful results. We saw that in the knockout samples, and you can see on the bottom here, we saw accumulation of MTA molecule in a variety of different tissue sections. What we also saw was that this accumulation only occurred in the tumor cells, right? In the region where we saw high tumor cell concentration. And that's because the normal tissue has the intact 9p21 loci. So we're not losing that ability to produce MTAP in the normal tissue region, but we are losing that ability within the tumor tissue. And we saw that for the wild type tissues that are MTAP positive, we were able to also detect MTA, but at a much less abundance. So one of the really interesting things that happened though during this process, that this paper came out from a different group at MD Anderson, where they said that they did not see elevation of MTA in these tissues. And when we looked at their methods, we realized that they were getting the bulk tissue and homogenizing and doing LC-MS. So they were mixing in these normal cells that do have MTAP expression. So because of that, we believe that they did not see this accumulation in the tumor tissue that had the MTAP deletion or the 9p21 loss. And you can see this very clearly in our mass spectra. 298 is MTA that we're detecting at a signal to noise of 18 in the sample that has the MTAP loss. And in the MTAP positive sample, we detected at a much lower signal to noise ratio, so a much lower relative abundance. And what really is neat about here in this data, you can see that there's this other peak at 298.074 that using high resolution mass spectrometry, we can separate these peaks and identify these ions. But if we weren't using high resolution, they would be combined together in the same peak and we wouldn't be able to see this trend. So we quantified this, relatively quantified these in tumor tissues and the wild type and knockout, and we did see a significant increase of this metabolite in the tumor tissue of the samples with the MTAP loss. We also saw a variety of different other metabolites that are in the methionine cycle, other intermediates that we can detect with DeBakey as well. So you can see here a variety of polyamines, spermine, spermidine, and then SAM and MTA. And now we can look more holistically at what are these pathways, what are the changes that are happening, and can we use this information to improve care for the patients. So we started this last year to work with the patient tissues. We are focused on bladder cancer for this project, which is one of the cancers that has the high frequency of 9p21 loss. We saw very similar trends so far in the human data as well, where you can see MTA in high abundance in the MTAP loss tissues compared to MTAP positive tissues. And we're continuing to study now doing Western blot and also looking at the whole exome sequencing to know where this 9p21 loss is occurring for these different samples in a variety of different tumors. Just so to give you an idea, this is the kind of data sets that we're working at with DeBakey and with the molecular data that we get. So it's a lot to process, and this is a very exciting project because we can now correlate this detection of a small metabolite with potentially response to immunotherapy. Kind of in the same theme, I just wanted to tell you about a project that we have been pursuing with Merck. We published a first paper on this in 2023, and we're continuing now to work on this project also as part of the Cancer Moonshot program at MD Anderson. Here we're looking at how are the tumors adapting in terms of their metabolite and lipid profiles after treatment with anti-PD-1 therapy. For this project, we started with preclinical models that Merck provided to us and with models that had different response to immunotherapy. And we started to look like as a function of time, as you're treating these tissues with anti-PD-1 therapy, what are the molecules that are changing? Is there anything from the metabolic or the lipid profiles that are indicative of a response to therapy? And what are these specific molecules and can we characterize them? So this is a typical analysis that we do as a function of time. And I'd love to talk more to Rob about the time-related statistical analysis of this data. But we saw that there was a change in the expression of specific lipid species as these animals were being treated and started to respond to immunotherapy. In particular, we saw that glycerophosphoglycerols, which are labeled there at the blue dots, were being weighted towards the treated tissues for the responsive tumors at day 15. And when we looked at this class of molecule in particular, what we noticed was that a lot of these glycerophosphoglycerols that were significantly changing in these tissues presented a large number of carbons. So they had longer fatty acid chain lengths, and they also had a higher number of double bonds. So these were polyunsaturated lipids that were accumulating in these tissues as we were treating these animals with anti-PD-1 therapy. One thing that is really valuable about our data, though, is that because we're doing spatial imaging of these metabolites, we can really look at the tissue histology and see, is this really homogeneously distributed in the tissue? Is there any specific accumulation? What's really happening on these tissues, especially for this class of PGs that we saw these polyunsaturated fatty acids? And what we noticed was that the distribution of these PGs, like for example, that 48, which is a polyunsaturated fatty acids, weren't really matching with the distribution of the cancer cells that the pathologists had been annotating for us. So we thought, okay, what does that have to do with the immune infiltration cells? Like what is really happening there? So we did the DESI imaging, and then we used the exact same tissue section, and we stained for CD45 and CD3. And what we actually realized was that these polyunsaturated fatty acids are overlapping with the distribution of the immune cells that are in these tumors, much more than the cancer cells themselves. So we're now in a phase where we're starting to work with our colleagues at MD Anderson to also get these samples as well from the patients and see, do we see also these changes in polyunsaturated fatty acids? What is it that's going on in the immune cells? Can we characterize this further and try to understand these responsive tumors versus the non-responsive tumors and how these lipids are playing a role in these mechanisms? I wanted to jump to another project very quickly, also with our colleagues at MD Anderson. This is a project that we're looking at anti-VEGF antibody therapy and how patients respond or not to anti-VEGF therapy in ovarian cancer. Anti-VEGF is being tested now as a adjunct therapy together with chemotherapy for patients with advanced disease in ovarian cancer. Dr. Sood, which is one of my collaborators, he's very interested in the role of the stroma, the connective tissue and the tissue around the tumor, and how these compartments of the tissue play a role in the resistance and the recurrence for these patients. And some of these tissues have been incredibly heterogeneous, as you can see here, with a variety of different histology. And with DESI, in the imaging assay, we can really focus on specific tissue areas and extract that data for molecular analysis. One of the really cool things that we saw in these results was a variety of changes, again, in the many different lipid classes in the treated tissues compared to control. And one of the very interesting results that we had actually related to the glutamate to glutamine ratio in these resistant versus sensitive tumors, where we saw, I'll call it GGR, a much higher GGR ratio for the resistant tumors when compared to the sensitive tumors. And we looked at that very carefully. You'll see here a variety of different ion images for many different metabolites in the expression of these molecules in specific tumor regions. Now, because of this finding, Dr. Sood started a clinical trial now that they're using glutaminase inhibitors as part of the treatment options for the patients with advanced ovarian cancer. So this is really exciting to us because one of the findings that we had from our DESI imaging study has really allowed us now to do this different treatment option for patients and test it out in the clinic. Now, the GGR ratio has been of really high interest to us, and this is another project that we started to explore with Rob and his student Aaron. Because we saw these changes in the GGR in the ovarian tumors, we started to think, are there other tissues and other cancers that this glutamate to glutamine ratio and that glutaminolysis is playing a role in the aggressiveness of these tumors, or maybe the molecular subtype of these tumors? And one of the cool things that we can do with our data sets is, because we're doing untargeted analysis of these metabolites and lipids, we can go back to the data that we already acquired and then start to ask more specific questions like, what about glutamine? What about glutamate? What about glucose? What about specific lipid species? Are they changing? And kind of like a retrospective analysis of our own data. So we decided to look into breast cancer because we had a large data set for breast cancer that we had the molecular subtype of the tumors that we had analyzed. And we thought, can we use this GGR to stratify these patients if there is any correlation with GGR in the different types of breast tumors? So this is what we did. We went back to our data using high resolution mass spectrometry. Glutamine is detected at 145, glutamate is at 146. And Kezia, my student, and Aaron went through this entire data set. We had thousands of pixels and data in mass spectra for both normal breast and breast cancer. Then, as you can see here, for breast cancer, we had the major molecular subtypes for these tumors. So we look within all of our data sets and we find these two peaks, and then we can extract this data for these two particular metabolites and calculate their ratio. And we did find a correlation that we're still exploring. So we're very excited about these results. With both DESI data and the mass spec pen data, we saw an increase in the GGR value in cancer tissues. And in relationship to the molecular subtypes, we also saw that going from like a lumino A and lumino B, we have a lot of GGR ratio, and then HER2 has a higher GGR ratio, and the triple negative has an even higher, yeah, Rob, GGR ratio. I don't think so. Prostate cancer was, I see Jeff here. Prostate cancer was not glutamate or glutamine.
It's glucose to citrate.
Glucose to citrate. Yeah, but I think it's fascinating because also in Graham Cook's group, we were looking at 2-hydroxyglutarate to NAA. So there are a variety of like metabolic ratios that we can actually measure. And even though it's not quantitative analysis, that level and that ratio is very indicative, even of molecular subtypes in this case. That's right. That's correct. Yeah, so it does like address the issue that with the direct mass spectrometry techniques is not quantitative analysis. So the ratio we can really look like at that rate of that particular reaction in the products that are being formed. So we're really excited about this project. We're now testing it in clinic. We're testing it with fine needle aspiration biopsies and seeing if we can use that data from the FNA to also get an understanding of what's the molecular subtype of these tumors. Okay, I have about 20 minutes and I'm gonna jump and change gears and talk about the mass spec pen technology. The mass spec pen technology was developed in my lab when I was still at UT, and of course now we've brought it to the Texas Medical Center and have several clinical studies that are ongoing. The process of tissue analysis with the mass spec pen was largely inspired by DeBakey. So when I was a PhD student and working with DeBakey, you know, the main observation that we had was that we can use solvents to extract molecules from tissues. Yes, Rob? Yeah, I'll get there. Yeah. So wait, where was I? So we knew that we can use solvents to extract molecules from tissue. But in the process of being in the operating room and being in the frozen room primarily, waiting for tissues, I started to think it would be incredible if we could repackage what we know already that works, which is using solvents to extract molecule, into something that was more like a device, like a gadget, something that a surgeon could just hold and use it and not think that they're doing mass spectrometry, not think that they're doing chemistry at all. And this is what we came up with the mass spec pen technology. So the goal was to make a simple interface between a very sophisticated instrument, which is a mass spectrometer, and what I call the inexpert medical user in terms of not being an analytical chemist or an expert in mass spectrometry. The other goal that I had with the mass spec pen technology was to make it complementary and not disruptive to the clinical tissue workflow. So one of the major goals was that the analysis would be gentle and non-disruptive to the tissue histology. So the mass spec pen, the way that it works is very simple. You'll see here, we actually 3D print the whole device. The pen tip, we 3D print the mold, and Maria was asking me this, and then we cast it with PDMS. And we like using PDMS because the biocompatible polymer is approved in several medical devices. We autoclave all of our pens before use, and then they're ready for our surgeons to implement in surgery. So at the pen tip, the way that we design is we have 3 channels. They meet at this reservoir. We can play with the dimension of this reservoir depending on the use, depending on the surgical procedure. Because we 3D print them, we've gone down to 500 micrometers and we've gone up to about 10 millimeters, so 1 centimeter. So once we activate the system, we fill this reservoir with this droplet of solution. And most of the times we're using sterile water. We actually grab the bottle from the OR. And then the water will interact, that droplet interacts with the tissue surface, and we extract small molecules, just like with DeBakey. We're looking at metabolites, fatty acids, and lipids. And then we open a suction system, and then we suction this droplet to a heated interface that's on a mass spectrometer, and that vaporizes the molecules. They go into gas phase, and they're analyzed by mass spectrometry. And like I said, one of the exciting things about this technology is that from a histologic perspective, we're not causing any disruption or destruction to the tissue. We've worked with tens of pathologists that have looked at the tissue before and after, and they're unable to tell that we've deposited a droplet of water, which you would expect, right? You just deposit a droplet of water, and then you extract it. So we can still preserve the tissue histology and do H&E staining and any other assay that the pathologists or the surgeons will already do with the tissue. In terms of the data, we get beautiful molecular profiles, very similar to DeBakey and electrospray-like analysis of small molecules. This is 3 examples from breast tissue. So on the top is a mass spectra of normal breast that we acquired with the mass spec pen. and then brass ductal carcinoma in situ, and then brass invasive ductal carcinoma. And what we've seen like across the field really of lipid and metabolite analysis is that there are really reproducible changes in the intensity of lipids. It's really rare that we find like one lipid marker. Primarily, we're looking at how the levels of these lipids are changing. And we found it to be very reproducible and characteristic of disease type, histologic types, and also molecular subtypes you can see here for these tumors. So we worked very closely with Rob and his team in developing statistical classifiers. We love using the lasso method that Rob developed because the output is a model that is highly interpretable. We get the sparse set of features that we can, we know that are strong predictors for whatever classes we're training our classifier to identify. But we can also then go and characterize these molecules, just like I said, we do with DESI, and know exactly what they are. What are these lipids? What is the fatty acid chain composition, the double bonds, and so on. So we get a classifier that's very robust, but we also get these markers that are highly predictive and that we can characterize and start to understand what's happening in these tissues. So the goal of the analysis really is to be very user-friendly. So we developed this graphical user interface just to show you how the process works. The user touches the pan on the tissue. We do the chemical extraction. That takes about 3 seconds. The droplet goes to the mass spectrometer. Then we use the classifiers that we developed with Rob to look through the data and classify the mass spectra as either normal or cancer tissue. And we can also display the probability if we wish to do so. We do not use this in the OR, just to note, because of IRB requirements, but we have the capability to do so. So we use the mass spec pan in a variety of different cancer types and tissue types. I'll show you a little bit of each one of these diseases, but overall, the process has been non-destructive to all of the tissues that we've analyzed. We're now actually, I don't have it in this graph, but we're looking at sarcomas as well, soft end bone sarcomas, which I'm really excited about. The analysis is generally rapid, anywhere from 5 seconds to 20 seconds, I would say. And that's primarily the time that it takes for us to transport the droplet in the tube. And we've kept an accuracy of about 96%. It really depends on the tumor type. Some, like for pancreas, we use like a 99% accuracy. For some other tumors like ovarian, we're like 92%. So it depends on the disease type and how different the mass spectra is. Cancer versus normal. For ovarian, we're doing subtypes of cancers, and for breast as well. So it normally drops when we're looking at different subtypes. And it may just be because we have less samples, right, for each one of the subtypes. So we made this animation in 2017 when we published the first paper, what we wanted it to look like. And at that time, we didn't have the system fully developed in the OR. We would have the pen coupled to the mass spectrometer in the OR. The surgeon would be performing a procedure. If there's a tissue or a particular region that they had a question, what is this, cancer or normal? They would touch the pan onto the tissue. We use an Arduino system, a microcontroller, to control all the steps. But once the surgeon presses the foot pedal, we deliver the droplet, extract the molecules, and then we open the suction of the mass spectrometer. That droplet gets transported through the tubing system to this heated interface that vaporizes the molecules into gas phase, and then you get these molecular profiles. And the thought was to make it as plastic, disposable. We of course sterilize all of our pans prior to use, and then we can wash it and sterilize and use it again, but we tend to normally toss them and make more. So we took this to the OR in the end of 2018. It was really quick going from the lab to the OR. It was also pretty nerve-wracking at first, but exciting as well. Dr. Silverberg was the very first surgeon that used our system. At that time, this was the mass spectrometer that we had. It's really big, like it's almost like it would sit in this table here. So it's a big Q exactive mass spectrometer. And we put it in the OR and we thought, okay, this is what we have. I really want to test it out. I really wanted the surgeons to test it out and for them to do the analysis and to give us feedback. Okay, this is good, this is bad. What do we need to improve? So just a few observations. The mass spec pan analysis was independent on the geometry or how rigid the tissue was. We didn't see any tissue, any limitations in terms of the tissue histology. We worked again with several pathologists. We would analyze with the mass spec pen and send it to frozen. Never had a case where a pathologist said, I can't read this because you did this analysis. They didn't even know we'd done the analysis. We've had no reported adverse effects, which of course is always something that you looking forward to. We sterilized all of our pans. We used sterile water from the OR, so everything was sterile, stayed within the sterile field. And my team, we were close to the mass spectrometer and doing the analysis in the mass spec. And we've now acquired thousands of mass spectra in vivo. And again, these are data that are being acquired by the surgeons. We're there to support, but it's really the surgeons that are driving the decisions, where they want to analyze, and they do the actual chemical analysis and the mass spectra data acquisition. So what medical decisions can we help with? Just want to play this clip from Grey's Anatomy. And if you were in my talk yesterday, you already saw it, but I think it's just really fun to see how it could help in patient care.
Okay, it looks like the tumor is free of the brachial plexus. Should we try to pull it out?
Let's do it. We've lost all the signal from one arm.
The tumor is moving pretty easily. Check your machine, could be artifact.
No, there's still a little tension from my side. I think it's coming from the root.
Which means there's no way to remove this without
Risking paralysis.
If they can't remove the whole tumor, they're gonna have to do it piecemeal.
But then they risk leaving some of the tumor behind. What?
My path in. Remove the tumor piecemeal and then use the pen to see if you got it all. Okay, it hasn't cleared FDA process yet. And it isn't on the market, but I'm telling you, it works.
I really have always liked you, Richard.
No, you haven't.
Yes, get it.
How can there still be cancer there? We've been at this for hours. It's still lighting up like Christmas.
Let's just keep going. We'll hit clear tissue eventually.
Signals fade. Really fun to see that. We actually have our real life Grey's Anatomy. We've been using intraoperative the technology now for several years. We had to stop our study for almost 2 years because of COVID. But now we're back at MD Anderson, Johns Hopkins, and Baylor. Yes, Rob. They didn't. No, I wish they would, but they just called me. We spoke for 30 minutes. They disappeared for a whole year. And the day before it aired, someone sent me an email. It's going to be in tomorrow. And I'm like, ah. So yeah, it's kind of exciting. There are several uses. I want to go quick to respect your time. We've been using it for tissue sensing and identification. I like using the word sensing because the way that I see it is that if a surgeon does need help in identifying a tissue structure, and we see that quite often with nerves and vessels, and also in parathyroid surgeries, identifying if it's a parathyroid or the lymph node or substructures of the thyroid. For a surgeon, it's really important to have that reassurance of what's being analyzed and what's going to be removed. So with the mass spec pen, we're able to get that identification prior to removal and really preserve the tissue in case it's not the tissue that indeed needs to be removed. We have several uses in surgical margin evaluation. You're seeing here it being used in the lumpectomy procedure, and also looking at cancer metastasis detection, and that's with in ovarian cancer as well. So just some example, this is all data that was acquired by surgeons. We did not acquire this ourselves. These were all of our colleagues analyzing thyroid, parathyroid, lymph node, breast, pancreas, and bile duct in vivo. Very similar profiles as we saw ex vivo. We did see some variations, but the same classes of molecules, same markers that we see ex vivo, we see in vivo as well. I get asked a lot about the effect of things like blood on tissue. Of course, the surgeons are doing everything they can to stop blood flow. I know we have some surgeons here. So there's not like, I think there's some misconception from scientists that there's like blood everywhere in a pool of blood, but there's always residual blood on the tissue. And we did see in the OR a much higher relative abundance of heme and hemoglobin. Now, because we're doing high resolution mass spectrometry, these peaks do not interfere with the markers that are characteristic of tissue. We still detect all of our small metabolites, fatty acids, and glycerophospholipids. So it's there, but it's not interfering with our analysis. We do everything that we can to avoid saline. salt is not good for a mass spectrometer. So we asked our surgeons to irrigate tissue with water, and they've been doing that. And this is how the pin looks like after use. So pancreatic cancer was something that I started looking here at Stanford with Rob, with George Potsides and Dick there when I was a postdoc here. And we went back to pancreatic cancer. This is a paper that we published in 2021. If you want to take a look, this is all intraoperative results with George Van Buren and Willem Fisher, the majority of these Whipple procedures. We were very careful here at looking at several different margins and looking at the agreement with histopathology. And in this project, we actually incorporated some of the operating room data into our statistical classifiers to see how that would improve or not performance. And these are some really exciting results that we have, and we're continuing to validate this intraoperatively. And thyroidectomies and parathyroidectomies, these are not oncologic procedures. These are for benign conditions that were used to identify these tissues. Our paper just came out in JAMA Surgery if you want to check it out. We have about 92.4% accuracy intraoperatively. Yeah, Rob. Excuse me? For parathyroid and thyroid is pretty perfect, but it's done just ex vivo. So let me jump there. For example, in this case, Dr. Sillerberg, my colleague, he removed a piece of tissue, certain it was normal parathyroid. We did our analysis. Thyroid and parathyroid are very distinct molecularly. So I was there for this case. I knew it immediately that it was not parathyroid. It was the thyroid tissue. Then he sent it to Frozen because he wanted to be sure. And then Frozen came back and it was actually benign thyroid. So the goal was to remove a parathyroid. He removed part of the thyroid. He had to go back and remove the actual parathyroid. So this is just to show if we had the mass spec pen in vivo, we could do that analysis non-destructively and guide the excision and preserve the tissue that's not intended to be resected to be removed. At MD Anderson, we're looking at advanced ovarian cancer patients. These are very difficult surgical procedures. These patients have advanced disease, so metastatic. They have undergone 5 to 6 rounds of chemotherapy preoperatively. So coming into these surgeries, the surgeons are trying to identify lesions that may be metastatic cancer, or it could be scar tissue or inflammation, and the ability to identify if it's really metastasis is very important. So this is not for surgical margin, it's primarily to identify metastasis within the peritoneum of the patients. So this is the PI of the project, Dr. Fleming, analyzing ovarian tissue ex vivo and then in vivo. And you see there our newer system. This is the Norbitrap Exploris and a rolling cart. We actually run this system in batteries. So you'll see here my student Mike. We just unplug it. It runs in battery. The mass spectrometer is still running. We roll it in. We roll it out of DOR. You have a little video here that we're pulling out of DOR. We take it to another operating room. We plug it in again. And we've seen very little effect on the performance of the mass spectrometer. You see it's very stable if we're acquiring data while it's moving. And we even went in the elevator, and we went in and up and down the elevator 5 times, and we saw no impact at all in the mass spectrometer performance. So we can acquire mass spec pen data using the power line, using the battery power. And this is just to show that mass spectrometry can be designed to be a medical technology. I firmly believe this. Now, there's a lot of skepticism. Mass specs don't belong in the OR. I would argue the opposite. We can design them as surgical carts and surgical systems that would run like ultrasound machines, and they can come in the OR and help surgeons make better decisions in the treatment of their patients. It's been really fun with the mass spec pen. We've done a lot of like prototype development. This is Jelling, my first postdoc, who's now left the lab, but he's made incredible contributions to this project. Our current version has this really cute graph per user interface and a touch screen that you can plug in all of your methods, and has the heated chamber that's helped a lot with cross-contamination. And this is the system that we use in the OR. Just to show the system can be miniaturized. This is a system that we have that we co-developed with Intuitive Surgical for the Da Vinci Xi. So it's a drop-in probe that's grasped by the robot to do analysis in vivo. And we're also developing a bronchoscopy probe. It's about 2 millimeters for bronchoscopy procedures. So we're very excited about that as well. So I talked a bunch, and I apologize. So much information, but I get really excited about sharing all that we're doing. And I want to thank primarily my collaborators in the clinic, all of the surgeons that believe in new technology, that are excited about trying new things and taking for their time for their incredibly busy schedules. I'm in awe of surgeons and how much time they're standing up, first of all, doing very complicated cases, and they still have the passion to work with us. So I'm very thankful to all of them. Very thankful for our funding that has allowed us to do these projects. And again, thank you, Sharon, for the opportunity, and happy to take any questions. Thank you. Oh, there's like a mass spec in here.
So I'm going to pass the microphone for a question so that those online can hear.
Thanks. I found the application and the translational components really exciting. So thanks for the talk. Do you need the same resolution as when you're studying, when you're training your models in order to attain the same diagnostic performance, or can you give up some of the resolution?
You can. That's such a good question. Something that we started to do now is we're using our DESI data to build classifiers, and we test it with the mass spec pen data. And then we get the mass spec pen, we build classifiers, we test with DESI, then we're also mixing it all. Because the nature of the data is the same. It's like solvents extracting molecules from tissue. So I think it doesn't really matter the resolution as long as we know the histologic composition. So if you're mixing in like necrotic cells with tumor cells with stroma cells, then we need to consider that. But as long as we know exactly where the data is coming from, we could, yeah.
So with DESI, you get a few hundred pixels per patient, right? Is that right?
Yeah.
But the mass spec pen is just one measurement typically?
So typically, it depends on the tissue size and the pen size. For the 2 millimeter probe, you know, if you have a little piece of tissue, we do like 3-4 measurements. If it's a big chunk, we do like 10 measurements.
But you never get much more than 10, is that right? You don't?
No, rarely. And some of the lung procedures we're doing now at the stapled margin, we get like 10 measurements sequentially. Yeah.
Thank you for the talk. I have a couple questions, mostly technical on the mass spec sample analysis and preparations. You say like it is more like when you do like DESI or mass spec MS pen, you don't dissociate any tissues.
No.
Okay, so how would you deal with all the extracellular matrix of the tissue itself, or is that a part of the contaminations that you kind of like you normalize to or delete it? Yeah.
So the way that I think about it, and I wish I had like a tiny microscope that could look at the lipids as they're being extracted, right? But I see the cell, like a tissue is like, it's not a pixel, it's a voxel, right? It's like a 3-dimensional structure. And I think about it like it as a sponge. And we have, you know, the cytoskeleton in the cells, and you have the lipids and the membrane, and it's fluid, right? It's all in water, it's liquid. So once we put that solvent on top, what I think happens is we extract like these molecules from the cells to the droplet. And the tissue itself, I think there's a remodeling in the cellular membrane if it's in vivo, or you're just really removing those molecules. But if you do H&E staining after, the cells are intact. If we do RNA analysis, the RNA is we've done like with DESI and no DESI, the RNA is unmodified. It's like we've not removed any of the genetic material. So I think we're gently pulling out these molecules, but the cells, they're still there. And we've proven that with like the staining technologies, like the proteins are still there. We're still staining for all of these proteins, and the mitochondria is there. So I think it's really just a very gentle process of using a solvent to pull out these molecules that are very fluid, right? We're mostly pulling out these very tiny molecules that they like to float around.
Right.
Yeah.
Thank you. And the next question is that I was wondering about like different sections of tissues, and also you have different tissues from different patients, I assume. How would you normalize it? Is there any kind of like signal to noise ratio that I need to be aware of? Let's say, because probably the ratio will be like very low to be differentiated. So how would you do that normalization?
Yeah. So I skipped some of the slides where we worked really hard in like establishing what we call quality metrics.
Right.
A lot of the times the surgeons don't really have any training, like they barely touch the tissue, or the extraction doesn't work well, or the droplet it kind of disperses. So there is some part of the technical aspect to get good quality data. So we've established some of these metrics where we say from like a data quality perspective, we have to meet a threshold of a signal to noise at least 5 for molecules XYZ in order for it to be predicted on. Now, from a patient to patient, it's remarkable how these profiles are reproducible. It is rare that if we have like a ductal carcinoma in situ for 2 patients, you know, we've even looked at like the effect of like being white or black or Hispanic, and the data seems to be very characteristic of the cell type itself more than some of these other factors. But so these profiles seem to be very robust and reproducible. So we haven't really seen a lot of like patient-patient variation as long as it's like in the same histologic molecular subtype.
Okay, thank you.
So I understand there's a question from someone on Zoom. Dan Cap, please go ahead. Dan, can you hear us?
Can you hear me now?
Yes, great, thank you.
Thank you. Excellent talk.
Thank you.
Intraoperatively, can you, without destroying the tissue, separate out benign ovarian tumors or masses from malignant ones and separate out the major subtypes of epithelial cancers?
So we've done benign versus malignant. We have a paper on that. We haven't tested it in vivo because our IRB, this clinical study, is only for advanced cases, and they're all high-grade serous ovarian cancers. But we have data showing that it could be done in distinguishing benign and malignant. And I don't think we've looked at different types of epithelial tumors, but I don't see why we wouldn't be able to do that and probably potentially discriminate them.
I think particularly if you can put this on a robotic arm, you can save patients some unnecessary oophorectomies by saying this is a benign lesion, don't have to do anything.
I see.
further.
Yeah, so that's what, you know, we would love to do more. We developed this prototype with Intuitive, and we haven't really done a lot of the robotic procedures. I know we've talked to some folks here in urology to do robotic prostatectomies, but also for like cancer screening, right, for detection of nodules and identification in like laparoscopy procedures, we could do that too with the drop-in probes.
Thank you. Very exciting work.
Thank you.
One more question.
Thanks. Great talk, Livia.
Thank you.
How about depth?
That's
So how about
good question.
if there's a little tissue sitting on top of tumor, does it give that signal or tumor signal?
That's definitely our major limitation, I would say. It's really like a surface sampling technique. We have tried to quantify depth so hard. Analytically, it's very difficult. So what we did was like, we got a little piece of like lung, and like we just cut this tiny section, we place it on top like an ovarian tumor, and we try to see if we could extract the data from underneath. And what we found is that like our depth, it depends on the tissue being analyzed. For like lung, we measure our depth to be about like 40 micrometers. So it's really like 1, 2, 3 cell layers. So if there's a tumor that's like buried underneath normal tissue, we wouldn't be able to identify it. It would probably be classified as normal, or improve systems to more deeply penetrate the tissue, or even invasively, like with a needle, go through a nodule and get to the core of the tumor or the bottom of the inside part of the tissue. But right now we're really looking at the kind of like, just like you see cancer on ink, right, for frozen, kind of like that outside layer of the cells and what's being detected there. Is that what you wanted to know?
It is, yeah. Thank you.
Yeah.
You mentioned using 1 to 6 probes per case.
Mm-hmm.
So why more than 1? What happens to them?
They get contaminated. So we have a flushing system between each analysis, and the way that we run it is we flush, and then we use our data algorithms to see, are we still detecting molecules from the previous analysis? And we can flush the majority of the, what we call like carryover. But if we start to see like accumulation of like blood on the pen tip, we normally tell the surgeon, let's use another pen. Or if we're going from like in breast, if we're going from like the distal margin, and then we go to a lymph node, with the surgeon normally picks up a different pan for a different tissue type. So we just lined them up on the tray thing with all the surgical instruments, and the surgeon has it right there.
Got it. Thank you.
So in your answer to my question, it was you only use maybe 5 or 10 measurements at most, but for things that are heterogeneous like the prostate, I thought one of the advantages of DESI was you get a lot of data points.
No,
and-
that's true. So with DESI imaging, you can cover a much higher area, but you also have to image, right? That
Yeah.
whole tissue area. With the mass spec pen, the thought was if you have the gland there and there's an area of the tissue that could have a metastasis or extracapsular like invasion or tumor at that surface, then you can analyze a few regions. And some of the things that we've done with our surgeons is for like the breast procedures are really hard because the margins are huge. So they will look at a margin that is closer to the tumor and then analyze a few positions. And if it's positive, we can go back and analyze a few more at a higher resolution. And we have the pens with the different diameters, but yeah, it's limited in terms of the area that you can analyze.
Thanks.
Okay, great. Let's thank Livia so much for that beautiful
Thank
talk.
you.